I'm in Andre Chalet's lab at UCSF, and we have, I think, a slightly different sort of set of problems. We're looking at much more structural and small scale. We kind of like to think of it as we're trying to go to systems biology from the bottom, in that we're trying to solve the pieces and then build up towards the system knowledge. The sorts of problems we've been interested in are these kind of assembly modeling and assembly dynamics problems. So we want to figure out the structure and motion and function of these structures in the cell. Some of which are a very small number of proteins, others of which are much larger. And we want to build these models that have, I feel like my volume's probably coming and going as I'm turning my head. We want to build these models such that they have the maximum possible resolution we can extract from the data, and this can be quite varied from things like the nuclear pore complex or the yeast genome, where it's very coarse scale. We know that sort of this large piece is kind of here and next to this other large piece, to much sort of higher resolution things like the 26S proteasome, where you have pseudoatomic models of many of the parts and pretty much how exactly things are and how they're relating. In order to do this, one of the challenges has been that we need to take lots of different data types and bring them together in order to solve this problem. It's not sufficient to take one type of experiment, build the model from it, and then and sort of present this model in isolation. So we've put a lot of work into being able to take sort of all these different types of data, combine them together. It is nice both from a biological perspective, in that we're being able to build models that simultaneously fit sort of as much as we can possibly gather, but also from the computational perspective, because having these different data types is often complementary and makes the optimization and other sorts of the solving the problem easier. To do this, we've developed kind of this four-stage formalization of this kind of this standard virtuous cycle. Everyone kind of references of you're gathering data, you're doing some computation, and you're going back into the and analyzing and then going back. And I should say, currently, this has generally been a very tight collaboration with experimental groups, since that's we do the computation, or other groups do the computation and another group does the experimental. And one of the goals, and I think something that could be very nice to come out of this meeting, are sorts of approaches and standardization, so these can be less tightly coupled, so the models are better shared. There's better incentives for putting the models out there in ways that other people can use them, and making them simple enough that experimental groups don't throw up their hands at trying to deal with the software we're producing. To give you a slightly more concrete idea of of the process, here's sort of a schematic of the process for two different systems, the spindle pole body and the 26S proteasome. The different things that are used for the proteasome only are in white, for the 26S only are in yellow, and the hashes are sort of shared. So there's a large range you can see of data going into it, ranging from kind of bioinformatics, statistical potentials on the sort of most, I don't know, some people would call it fuzziest, most theoretical side, and then various types of structural data, electron microscopy, protein connectivity from various sorts of proteomics data, FRET, things like that. Once we have some amount of data, we choose a representation for the system and encode the data as a scoring function. So each piece of data is some scoring function term, and then we have a system where minima of the scoring function represent solutions that fit all of our data. This is nice because we can then throw sort of a lot of optimization tools at it, ranging from Monte Carlo, as is used in the spindle pole body modeling, to discrete optimization protocols that we've developed internally. And then there's a lot of analysis, checking it against data that you haven't used, and typically repeating this over and over again. One issue I should also mention, as it is sort of relating to the subject of the meeting that we've encountered frequently, is one would like to be able to, when doing this process, incorporate data from the literature in addition to what your direct collaborators have gathered. And this has been a significant challenge for us for the sorts of issues that have been mentioned in the recent talks of quality control, non-standard conditions. You end up with, you don't know, how good the data is once it's out there in some paper, and there's errors and contradictory data, and trying to resolve these in a reasonable manner has been a challenge. So unfortunately, much of the modeling efforts, the 26S here being one notable exception, have depended almost entirely on sort of collaborator-gathered data as opposed to data from the literature. The 26S, it's worth noting, their proteomics data from the literature was gathered, but was used, but there had to be a fair amount of culling involved in that some of it just seems to be, wrong. I mean, not fit the model or any of the other models that have come out on this subject too. So to support these sorts of modeling efforts, we've developed this software we call IMP for Integrative Modeling Platform. It's structured in a bunch of different layers to try to be from sort of the most general to the most friendly. I would say right now, I guess as shown by the sizes of rectangles, much of the weight is down on the general programming side as opposed to the data file-driven sort of approaches. The sorts of tools that have been developed as part of this project, and just on the sort of higher level things, there's web apps and Chimera, which is one of the molecular viewers. Wow, I haven't been very good on time. So driven tools for doing SACs and things like that. To say, talk about the lower level, we've structured it as a set of kind of independent parts that you can mix and match in order to combine together to build tools to solve your particular systems. Everything is open source. It's structured as independent modules so that people can contribute their own code easily without having to sort of license it to us or distribute it with our software and things like that. And just to mention, to try to trigger a discussion for later, one big challenge we've run into is the sorts of things we're producing are these models. They're not necessarily atomic. They relate EM data, structures, coarse grain structures, things like that. And so representing this and visualizing this in an interactive way has been a big challenge. And we've had a working group going on with some people from Chimera, some people from the European PDB, to try to solve this problem and come up with tools for that. And I think it would be nice, this may be a good topic for discussing later. And then we'll just go. Oop. I was supposed to put up the acknowledgements, but since my time is up, I don't know why my acknowledgements don't seem to have made it anyway into the PowerPoint translation. So